on nematodes. And that'll probably be fairly good news to Dr. Tilke and Baum, but primarily because the impacts are dramatic. Obviously, it's a big deal in soybean production. And we're very concerned about how we can manage that pest. I shouldn't call it a pest, that organism, in terms of making sure we can keep soybeans productive. And obviously that the last few years, people have worked very hard to figure out what genes are being turned on by the organism, what resistance genes are important, et cetera. We're going to step back from that and assume that the organism has infected, and we'll look at the consequences, both in terms of things that we know are important for the way the plant determines yield. And that's why I put those grain yield equations up there. We can look at it from the perspective of what does the plant enabled in terms of water use? How much water can it get into the plant? Because this is a #1 issue. You may know that the vast majority of water that plants take up, it simply blows right through them. It's for transpiration. It keeps them cool, brings up nutrients, et cetera. A little fraction of it goes for growth. Well, if the organism is impacting the way water uptake occurs, or the capacity for water uptake and the transfer up to the shoot, well, that's not a good thing. And we'll talk a little bit about that. The other big issue, of course, is how plants convert electromagnetic radiation energy, sunlight, into chemical energy. That's what they do. And that's why we find them so valuable. And basically, it's just taking one form of energy and converting it into another. And we're, of course, very concerned about what they do for making chemicals that are valuable to us. Biomass is an important chemical, if you want to look at it that way. And if we're concerned about biomass, if we're looking for an energy crop, then of course biomass per unit energy is really important. If we are concerned about taking seeds for the next generation or for chemical use or whatever we want to use them for, then of course we need to worry about a harvest index, how the plant partitions that energy into what we want to harvest. The thing that many researchers, for some reason, fail to recognize is that the formation of yield is determined not only by how much grain comes off the field, it's how many grains per plant, how the plants decide how many grains to add to their system, to their mass, and of course, how much they fill once those seeds are formed. So that's why I put in the grain weight equation with those two components, grain number and grain weight, because we'll look at those things. And it's pretty clear that soybean cyst nematode, which we'll talk about, has a very large impact on the formation of seeds as well as their development. So we'll touch on those things as well. But again, from the context of how plants function, how the roots and the shoots function together. All right, well, you know this organism pretty well. It does not do good things in terms of when it infects a plant. It doesn't kill the plant. It is a kind of a saprophytic organism in that it develops those feeding sites called syncytia. And it draws nutrients and water, mostly nutrients and water from the phloem, sapping what could be going to the roots. And that's something we have to kind of delve into is so what? How big a deal is that in terms of maintaining overall plant function, seed formation, seed development? So we'll look at the inhibition of plant growth, plant senescence, things like that over the next few minutes. I had to bring this little piece of data up from a nice article recently out of Greg Tilke's operation, because it clearly shows that developing resistance to SCN infection is controlled by the genetics of the plant. Obviously, genetics is really important, and that's why there's so much energy being spent in terms of identifying resistant germplasm and the genes that are controlling that resistance. What's interesting also beyond that is the variation that shows in the right side of this figure, which is a number of successful reproductive products that the soybean cyst nematode produces. You'll notice there's not much of a correlation between how many eggs, let's just call it, how many eggs are produced, which means how successful the organism was in infecting the plant, and the yield variation. So there clearly is an environmental component to the impact of that organism on the way the plant functions. So we want to talk about that as well. Unfortunately, we don't have a control of that. We really don't know how to manage that environmental variation. So what's the recommendation? I think you all know it very well, that find resistance and use it as best you can in some kind of rotation, mixing the 188, 788 resistance with the Peking resistance. Otherwise, you're going to have a lot of eggs forming on those plants. And we'll see what happens to root growth when that variation takes off. I think it's important to recognize that the organism, soybean cyst nematode, is one that once it invades the plant, it forms a feeding site that doesn't damage the vascular tissue. I was kind of surprised by that. I thought those synthesia forming, those cells that were modified around the vascular tissue that basically form transfer cells, a very large mass of transfer cells, that they would damage the vascular tissue, the xylem and the phloem, so that water transport would be disrupted and phloem transport would be disrupted. That's not the case. No, they are very clever in that they just get close enough and effective in terms of how they modify the cells for transfer of nutrients so they can draw nutrients out of that source. So that's the good news, I suppose. The bad news is, of course, they're very good at it and they draw a lot of nutrients, and we can see what impact that has on root development and the impact of what we're going to call the signals from those roots that have a large impact on plant development. Resistance, of course, has been identified in terms of how the plant, how it fends off the initial infection, in the sense that it doesn't allow the organism to form a feeding site. So if the organism can get a feeding site established, and the maintenance of that site is something that they're very good at, and that's when a lot of nutrients are drawn from the plant. So if you're looking for a way to fend off the organism, it's very important that you do it, you focus on that initial infection and establishment of that synthesia, the change in the way those cells are modified so that they draw nutrients out of the phloem. But that still isn't going to kill the plant. That's not what they're doing. Of course, that wouldn't be very logical, would it, to kill the plant that you're trying to feed on? So we have to think about what is it that's happening with this feeding that is changing the way the plant is developing, changing its capacity to handle water stress, handle a decrease in assimilate supply, photosynthesis, the way it forms seeds, all these kind of things we're going to be talking about in the future, or in the next few minutes. The backdrop for that discussion is that soybeans, they just have a pretty weak root system. It's kind of fibrous, it doesn't go that deep. It has a lot of resistance for water transport. So it's it's a background from which, if you change its capacity for doing what it ordinarily does, water transport, nutrient transport, sending signals to the shoot, then the environment's impact is even greater. So when water becomes limiting, if soybean cyst is around and has chewed on the roots or is sapping off the roots, then the environment looks all the more stressful to the plant. So it's not that hard to think about what this organism is doing when you recognize the fact that it's infecting a part of the plant that isn't very vigorous in the first place. So what happens? I mean, in terms of anticipated impact, I just listed 4 things. You can probably think of some more. But one of the things that the cyst does, it increases partitioning of assimilants to the roots under good conditions. In other words, if there's plenty of water, plenty of nutrients around, the plant will allocate more resources to the roots. Why? Because there's another sink down there that is drawing assimilate out of the phloem that isn't getting to the roots. It's being bypassed. So the plant will respond by sending more down there. It's amazing. It's not like the plant says, I can distinguish between the roots and the synthesia. It can't. So it's sending assimilates down. So what happens is there's a depletion of assimilates, that would ordinarily go to the roots. That's something we can assume will happen without even going down there to measure it. Decreased partitioning when photosynthesis is inhibited. This seems maybe counterintuitive, but the reality is when assimilate supply is limiting in the shoot is less generous, okay? The leaves aren't going to suffer nearly as much as the lower parts of the plant. The roots will indeed get less assimilates. Their proportion will decrease when the shoot is limited in its capacity to assimilate carbon from the atmosphere. So what's that mean under conditions when soybean cyst is there? Well, the roots will get even less. So stresses that inhibit photosynthesis become, again, even more dramatic. Because there's an inhibition of root development, root proliferation, the parts of the root that are sending or producing chemicals that are important for shoot function, that communication changes dramatically. So the root-derived signals that we know have an impact on photosynthesis, for example, and seed formation, seed development, those signals are altered. And that has a big impact on the success of the plant reproductive development. So all in all, what that means is if there's a difficult environment, it's going to look all the more dramatic to the plant. So let's see if we can follow up on that. Well, some examples. I don't know if you remember the yields project for years ago. I kind of dusted these data off to show you. This is a comparison of a cyst-resistant variety versus one that it was less or more susceptible. And basically what it shows there's a variation in just how stressful the impact of SCN is on the plant, all right? In the year 2002, it wasn't that big a deal. The resistant and susceptible, you couldn't tell them apart. But in another year, like 2000, it was a lot more dramatic. Well, why was that? Well, 2000 was a very stressful year relative to 2002. There was plenty of moisture. The plants, even though they may have had less root mass, and there was assimilate being diverted by the cyst, the soybean cyst, they managed just fine because they didn't use all their root system to capacity. In a year that's stressful, when root growth might be somewhat inhibited, and the impact of a altered allocation of resources to a cyst has a much bigger impact on the plant. So in the dry year, cyst is absolutely terrible. And you might not even see it if cyst numbers are growing, increasing in the soil in very good years. And on the right, what you see is the egg production. Interestingly enough, in a very good year, in a very healthy plant, when there's plenty of assimilate around, the soybean cyst thinks that's just the most wonderful thing, makes lots of eggs. So even when there's a lot of assimilate, doesn't seem to be a yield impact, I think you know the fact that the plant is allocating resources that it wouldn't ordinarily do to a soybean cyst and to get a lot of seeds produced, or a lot of eggs produced. We looked into this a little bit to try to figure out whether that's a shoot-derived or root-derived response, and it's clearly a root response. How do we know that? Well, we took shoots of soybean cyst nematode resistant plants, put them on roots of susceptible plants, and did the converse, took the susceptible shoot, put them on a resistant root, and you find that the resistance goes with the root, and the number of eggs that the soybean cyst produces over the course of this experiment is much greater in the plants that were growing very well. If root growth is inhibited, then soybean cyst numbers are down. So it's a bit of a paradox, folks, that the plant is very willing to allocate resources to that cyst. It doesn't control that in any way, shape, or form. So if we have a lot of root growth, a lot of shoot growth, good conditions, the soybean cyst is going to do very well. That's why, of course, it's so critical to find resistance so that a very healthy plant isn't feeding that organism. One of the things that we know happens is that photosynthesis decreases, the generation of carbon. And I had to admit, I had to dig pretty hard to find evidence of that. This article, I can't even see, what was it, from the mid-'80s, I guess. they use a tool that they can separate the gas exchange of the shoot from the gas exchange of the root. And they were most concerned about, is carbon production decreased by the shoot? And is the ability of the root to utilize that carbon changed? All right, so they had that cute little system on the right to do that. Well, the numbers are striking if you look at the table on the left, which I extracted from that paper. And the idea is to control to compare the capacity for photosynthesis, which is that PN column. There's roughly a 30, in this experiment, 32% reduction in the capacity to fix carbon on a leaf area basis. That's not the whole plant, that's leaf area basis, all right? That's a big reduction. The root had a almost 50% increase in respiration. What that means is that part of the plant, and of course it was infected in this case, was utilizing more assimilate and respiring it away much more rapidly. So the plant was clearly allocating resources to the root, and the presence of the cyst increased the amount that was used for respiration. So the efficiency of development, efficiency of growth clearly had to go down, and you can see it in the biomass. All right, leaf area down about 28%, shoot dry matter down almost 40%, and root dry matter down about 30% relative to the untreated or controlled plants. That's a very, very big hit in these small plants. I was shocked by how large an impact this had in plants that were growing for about 40 days, something along those lines. Not very long, not very long. They're vegetative plants. What concerned me the most when I saw this was the impact on root growth, because that is a very, very large reduction. And you know what's happening. There are fewer growing points. There's less capacity for taking water nutrients out of the soil, clearly. But the major issue I was wondering about and concerned about was, if there's fewer growing points, we know that there's going to be an impact on chemical signals coming from the roots. So root to shoot communication, we got to think about, is this going to be affected, or how much is it affected? Really, not is it affected, when the soybean cyst is altering the development of root structures. And the two hormones that we know are really important coming from these root apices are abscisic acid, ABA, and cytokinins. You might be familiar with ABA, of course. It's often considered an inhibitory type chemical in that it slows down growth, leaf growth, formation of new apices, things along these lines. It also closes stomata, regulates stomatal aperture. But it is a positive effector in the roots. Under stressful conditions, an increase in ABA actually maintains root growth. So that's kind of an interesting dichotomy. Cytokinins clearly are involved in regulating cell expansion, and that's going to be really important for setting seeds. And of course, cytokinins are clearly involved in the longevity of leaf tissues, regulating senescence. So we're going to talk a little bit about that. So you can see right away that where I was focusing where I was leading, where I was being led, I should say, to what's happening in as far as the impact of soybean cyst on root development. And of course, we got to look at ABA. ABA, we know, is one of many, many factors that have an impact on stomatal regulation. And of course, stomata, you know, are the pores in the leaves and other structures that allow gas exchange to occur. CO2 in, oxygen out, CO2 out in respiration, things along those lines. ABA is a very important regulator in the sense that it has a dominant impact on long-term regulation of stomatal aperture. It'll close stomata quickly, but it'll also keep them closed because it shuts down a lot of the internal workings that control ion movement in and out of the stomata, out of the guard cells. Anyway, I wanted to make sure you understood that ABA is coming from root apices. It's coming from the roots, and it's transported up to the shoot via the xylem. And under stressful conditions, ABA goes way up in the xylem, and it reaches the guard cells, and it will close them. And the consequence, immediate consequence, of course, is less gas exchange. And I'll show you an example of that in just a second. But I want to emphasize ahead of time that this soybean plant, its gas exchange, carbon in, is absolutely dependent on keeping the stomata open. It has no mechanism whatsoever for concentrating CO2 from the atmosphere. It's just got to diffuse in. So if the stomata are closed, you know what's happening. Gas exchange is slowing down. There was one article I found from some folks in Brazil that infected plants. It was Embrapa 133 soybeans. And you know about Embrapa? That's the folks down in Brazil doing the basic research, great organization. I don't know exactly what the SCN infection levels were. I think those are whole numbers. I can't say that for sure because I couldn't interpret it. But the bottom line is, when you look at the graph on the left, photosynthesis rate, the round circles are the controls, no infection. Everything that was infected had decreased photosynthesis rates. Everything, even the low rates. That will only happen if those stomata are getting a signal to close stomata. That's what's going to control. It's a totally stomatal regulation of photosynthesis. The other curve there talks about chlorophyll content is down. And of course, cytokinins coming from the root have a huge impact on maintaining chlorophyll content in leaves. So the folks didn't use this to talk about what's happening from the roots, but you don't have to have much of an imagination to figure out that that impact that they're having, those cysts were having, had to do with what was happening from the roots. And I just grabbed a couple more comparisons in from this paper. Leaf photosynthesis versus stomatal conductance. Of course, stomatal conductance decreasing, leaf photosynthesis goes down. Leaf area duration decreased versus plant dry weight. Yeah, of course, you cut down leaf duration and you're going to have smaller plants. Plant dry weight and grain dry weight, those are always going to be closely correlated. And of course, the root dry weight, decrease in root dry weight was closely correlated to grain dry weight. There's not one good thing on that table. I'll tell you that right now. All those limitations, stomatal conductance, decrease in photosynthesis, decrease in green leaf area, all that stuff is negative relative to the capacity of the plant to put on seeds. All right, can I get to the next one? Help me here. There we go. So that leads us to thinking about seed formation. One very important aspect of seed formation in this plant is it's progressive, all right? A lot of the flowers are produced but never make seeds, but all of them can, all right? And we know that signals from the roots are an important determinant of whether or not those flowers that initiate a pod will actually translate that initiation into a expanded structure and continue to put seeds on. So again, we're going to be looking at cytokinins. Why? Because there's a lot of information that says at this part of development, the structures are very much paying attention to cytokinin levels and the capacity for expansion. The little figure on the right is very, very, what would I call it? Basic anatomy, in the sense that you can see there's a very rapid transition from just producing the structure to expansion growth. That transition marks the success of whether or not an initiated pod is going to form seeds. All right, if that expansion does not occur, you're not making seeds. That expansion period is closely correlated, and I would dare say dependent on the flux of cytokinin from the roots. And this has been shown in several different ways. I've just got one example here where they actually measured how much cytokinin was coming up in the xylem, and it was right on top of the capacity for setting seeds. If you add cytokinin to the system, people have done this ad nauseum. I'll admit, I've done it. Spray cytokinin on these little flowers, you can increase their percent set. Easy, all right? Anybody can do this. It's not a mystery. So what the important part here is that the timing is critical. If you look at the figure on the right, it basically says in those positions on the rachis that have a high percentage of seed set, they get a lot of cytokinin, okay? So if you disrupt cytokinin coming at this time in development, you know what's going to happen. You're going to decrease the potential for forming seeds. All right, I'm not going to harp on that too much more, but it's pretty clear. So the only question is, where are these cytokinins coming from? I hope it's pretty obvious by now where they're coming from, all right? And if you disrupt that flux, you're going to decrease seed set. It's just that simple, all right? So we don't want to do that. The other piece of the puzzle, if you will, as I mentioned, comes from the association between assimilate supply and formation of the seeds. Excuse me. There's been all kinds of work on this sort of thing, focusing in on, seed supply or assimilate supply during the period when plants are setting seeds, this middle of the season. How important is that really? Are the plants, are they luxurious in assimilate supply? Are they tightly dependent upon it? What are they? Well, this figure takes away any guess or any question about it. Kernel number per plant versus plant growth rate. Soybeans, the relationship is linear, okay? The very small plants, they set a pod just as efficiently as a very large plant. That might not make any sense, but that's what they're doing. Why? Because the plant is structured as a series of phytomers. Every phytomer functions the same way. There's a leaf, there's a node, there's reproductive branches there. That leaf is feeding those branches. So if the growth rate of the plant is increased, there are more nodes, there's more productive sites, there's more assimilate available per site. It's pretty straightforward. So when you look at this little soybean plant on the right here, that is from, it was once, at one time it was the yield, what do they call that? Maximum yield winner, like 150 bushels of soybeans. You can see that that plant has lots and lots of nodes. You can see that each of those nodes are pretty well filled out with pods. I'm going to just ask you, guess what this particular grower pays attention to when he's growing his soybeans? What part of the plant is he mostly concerned about? Yeah, it's not the shoot. He is managing the roots, this guy. It's amazing. Kip Cullers, you might know him. So it tells you that maintaining a very healthy and vibrant root system has more benefits than simply make sure there's enough water and nutrients coming up from the plant. They're sending very important chemical signals to the shoot saying, you can continue to grow, expand, set seeds, set more branches, all that stuff that determines whether or not those initial structures will continue to form the seeds that we think are so important. When we look at this from a canopy perspective, it's a little bit of a different picture. All right, we know that soybean production is very much focused on closing the canopy early, making sure we intercept all the light and get the most out of that season we possibly can. Well, that's very good. We should be doing that. But you'll notice from this figure on the left, biomass versus intercepted PAR by Larry Purcell down there in Arkansas, that this canopy is saturated for light. It can't use all the light at high yield levels. What does that mean? It means that something else is limiting, all right? Limiting for getting CO2 into the leaf. The stomata aren't open enough. They can't diffuse enough CO2 in fast enough to use all the light. What do? They just re-radiate it. They don't use it all. So that's an interesting problem because the corn plant doesn't do that. It's got a CO2 concentrating mechanism that allows it to use all the light you can possibly give it. All right? It doesn't become limited for CO2. The soybean plant does. Yet, it's critically important to maintain the maximum amount of assimilation over the course of the season. And you notice that that previous curve was linear. During the seed filling period, the response to assimilate supply is absolutely linear. But over the course of the whole season, there can be what appears to be additional light available that the plant's not using. All right. So those who have looked at this kind of thing in the past asked a very simple question. How much does a soybean plant depend on assimilation over the course of the season relative to what's happening during flower and pod set? Well, if you take the integral into that curve, the soybean curve. You see that it's less than the corn curve because corn's more efficient at a given light intensity. But the integral of that curve, if you can modify that, if the soybean has something happening to it that decreases the size of that curve, whether it's early on, during flowering, or later on, what impact will that have on the overall productivity of that plant? Well, I'll tell you, it's pretty straightforward. You just don't see very many linear relationships in biology, okay? Now, these aren't really high yield levels in this particular figure, but it tells you very plainly that the yield formation process in this plant, seeds, setting seeds, filling those seeds, and if you're concerned about composition, you know, that's a assimilate supply issue as well. That anything that decreases the integral over the course of the season is going to have bad, it's negative. Even though that canopy is saturated for light, it can't use it all. So that's a real limitation in this crop. If somebody wants to earn a lot of money, turn this crop into a C4 plant, and this curve will look a lot different. And maybe there'll be less of an impact on things like soybean cyst drawing assimilates out of the root. The last issue, well, close to last issue, we'll deal with here is once those seeds are formed, the plant is remarkable in that it can continue to fill those seeds as long as the pod can continue to expand and the plant will continue to apply assimilate. There's no real stopping it. It's limited by more a morphological limitation of the pod than anything else. It's really interesting. You can pack more protein in the seed with more assimilate. You can make the seeds bigger with more assimilate. They're remarkably responsive. This figure draws, on the left, draws a lot of data from all kinds of literature. And it basically says the line going across there is a zero line, no response to a change in assimilate supply. On the left, if you decrease assimilate supply, in other words, shade it, stress it, soybean cyst it, whatever you want, you'll decrease the size of the seeds. Yeah, you can make smaller seeds pretty easily because they're paying attention to current assimilate supply. What is amazing about it is if you can add more assimilate, the plant responds in a very positive way. Grasses will not do that. Corn will not do that. Wheat will not do that. Once that size is set, you can't make them bigger. You can make them smaller, but you can't make them bigger. So that raises a bit of an issue when we look at the impact of soybean cyst on the senescence process, on the duration of seed filling. And that's what the figure on the right is all about. Okay, I think this one comes from Ohio State. I should probably give them a reference. Anyway, pretty obvious. We're looking at two different varieties there. One's resistant, one's susceptible. You don't have to use much imagination to figure out which one's susceptible. The duration of green leaf areas is shortened. All right, where do you think that's coming from? Okay, what's the consequence of SCN infection on hastening leaf senescence? Well, I've given you a lot of clues already. There's less root area, less root growth, less apices producing those chemical signals that maintain growth. You know that's going to lead to senescence. The plant can't stop it. Well, cytokinins coming from roots are right in the middle of this. They are the ones that are dominant. They're the hormone that's dominant in terms of controlling leaf senescence. Very nice article in the Annual Review of Plant Biology describes this in a lot of detail. But there's just plenty of evidence. Cytokinin levels drop, all right? If you can increase cytokinin content with the sprayer, I'll show you another example, molecular approach, you can delay senescence. That is easily done. The genes, if you follow those for cytokinin synthesis, cytokinin synthase and adenosine phosphate isopenyl transferase are downregulated during senescence. And the gene for cytokinin degradation, cytokinin oxidase, is upregulated. So it's pretty obvious what's happening. The plant has, once this process has started, decreased the capacity for cytokinin content to be maintained in those leaf structures, and they will continue to senesce. Other hormones are involved. There's a whole suite. It's a very complex process that we could talk about another time if you like, but cytokinins are at the absolute center of it. How do you know that? Well, you can increase cytokinin using a transgenic approach, and that's what this cute little picture at the bottom is about. That's tobacco. Of course, tobacco is pretty easy to transform. That's why they did it in tobacco. Plants on the left were stressed for a very long time, water stressed, and part of them were transformed for increased cytokinin production, others were not. And on the right, you can see what happens to those that can recover from a water stress. They don't lose their leaves. Pretty amazing. There are limits to this, of course. We want abscission to occur, and that might be something we have to pay attention to, obviously. But the message is clear. How can we minimize the impact of soybean cyst nematode on the function that the roots ordinarily have in terms of maintaining green leaf area? All right, so the question I'm going to leave you with in this particular part of the talk is, what if we could minimize the impact of a soybean cyst infection in plants that are losing their resistance, like in 188, 788, if we were to upregulate cytokinin production in the roots? I think that would be a remarkable little test and not hard to do. All right, well, just to kind of wrap it up, we were skimming quite a bit, all right? But I think I tried to focus on a few things that we know are critical. And even if we don't have perfect resistance available to us, there may be ways to modify the plants we have available to improve their resistance. But the problem centers around the fact that the plants have a pretty weak root system. It's probably not the right word to use, but it's a general word that just renders it vulnerable to difficult conditions. If those conditions, if that weakness is exaggerated, then that becomes even worse. So when synthesia form and they divert assimilates, they decrease root growth and the development of those apices, they just expose that weakness to a greater extent. So they make a difficult situation that much worse. We know those root apices are really important sources of signals to the shoot, and basically telling it, continue to grow rapidly, slow down in growth, or stop growing. Those signals are dominant. All the things that we talked about, stomatal inhibition of photosynthesis, decreased seed set, premature leaf senescence. I mean, it's pretty clear that those signals, that communication to the shoot has been altered by the presence of a soybean cyst infection. So hopefully we can overcome that if we're clever enough. And one example I've provided is, we know we can upregulate cytokinin production. That's not hard to do. But can we do it in a way in soybeans that minimizes the impact of soybean cyst? That would be kind of a fun thing to talk about. All right, I appreciate very much your attention. And of course, if we have time for questions, I have time for questions. I'm retired, I could spend all day here. So thank you very much.
All right, thanks, Mark, for the very interesting talk. I have unmuted everybody, so if you're muted, it's because you have yourself muted. So at this point, feel free to ask questions.
I have a question, Leonor, that was actually sent to me. But so if we're thinking about other pathogens, Mark, such as a pathogen that kills the root tissue, for example, Fusarium, how do you think that might affect the plant sending those photosynthesates down to the root? If the root's now rotted,
Yeah, it'll stop long before you see the rotting. If the cells are degraded and they're not actively utilizing assimilate, they're not sinks anymore. So what you see as a rotting is way past the, let's call it the decision to send assimilates down to the roots. Remember that the shoot does not control where, the source of assimilate doesn't control where they go, all right? The leaf produces assimilates and dumps it into the phloem. It has no control over after that. So it could go to the roots, it could go to anything, go to stem, whatever, depending on what's the strongest sink, what's utilizing assimilate most aggressively. So if the cells in the root have lost their capacity to utilize assimilate, they've long since not been a good sink. And the impact of that loss of root function has long since happened. Okay.
Yep.
Hey, Mark. This is Steve Whittam.
Hi, Steve.
Hey, I was wondering, you said the soybean has a weak root system. How would you improve it?
Yeah, I guess I don't have a really good answer for that, Steve. The value of the root system in terms of where it can attract assimilate kind of depends on the soil. So a dense root system, or forgive me, a heavy soil, clay soil, boy, it'd be really good to have a nice, big, heavy taproot, right? In a finer soil, lighter soil, a more fibrous root system. So there's lots of debate about what makes it for a better root system. And I guess we'd have to think about where we're going to deploy those plants in terms of what would make it better. I certainly would argue that whatever you do, that it be a fine enough root system that it has lots of growing apices, because that will protect the plant from loss of individual root fibers. Others can continue to provide the important signals to the shoot and regulate stomatal aperture, et cetera. So I'm kind of in favor of a more fibrous root system if we have plants that are generally growing in lighter soils.
You think structure of root system has been influenced by breeding over the years at all, or is this something that's not really been?
It has in other crops.
Mm-hmm.
If you look at work that Jonathan Lynch has been doing over the years at Penn State, I think, they have been very active in modifying the root system of phaseolus, so common beans, and it's been great. They've figured out that plants can be more efficient for phosphorus uptake, for example, if the roots are finer and longer. Water uptake is improved. The efficiency of carbon allocation is improved if they have less respiration, things along those lines. So there's a lot you can do if you're willing to do what Lynch calls shovelomics and actually look for morphological differences.
Okay. Yeah.
Mark, this is Alison again. And I was thinking about the slide you showed with the soybean from Kip Colors and how he manages the roots, and that therefore improves shoots and yield. And so there's a lot of talk at the moment about soil microbiome, and there's a lot of research going on in that area, looking at soil microbiome and what's going on there. And I just wondered what your thoughts were on, could we improve the soil microbiome to manage the roots of soybean better and therefore, improve yield on soybeans? I just wondered what your thoughts were along those lines. If you thought...
You're bringing up a sad day for me. I'll bring this up. I do have one regret in being a member of the agronomy department. And I guess I've shared this enough, I don't know. When I first came, I said, what we really need to work on? We need to figure out how to farm, if you will, or manage the root microbe systems so that we can improve root microbe interactions and improve productivity. I mean, what people are really doing when they fertilize is managing the microbiome. They're not fertilizing plants. And I got the most odd looks. So I kind of let that one go. And I should have followed my instincts and came over to your department and said, hey, this is the most wonderful opportunity. As you can see, people are going crazy with it right now.
Yep.
Everything from seed treatments to additives to the soil to looking at microbes that fix nitrogen. There's just so many things going on in the microbiome world now that it's fantastic. So I wish I'd done it. I apologize. But and if I were young like you, all these wonderful young faculty you have, I'd be on that. It is the future as far as I'm concerned, as far as managing crops.
So Mark, this is Rajiv.
Hi, Rajiv.
Good to see you. Quick question. So these resistant varieties, of soybean that are resistant to this cyst nematode, do they happen to have stronger root system than those that are susceptible?
Interestingly enough, what little work I've done on that, when we compared resistant and susceptible varieties, their root systems were just the same. We couldn't really tell them apart in terms of their structure and mass. Of course, that was in a chamber, so I've got a caveat there. But no, they're not different from what I have seen. And my experience, unfortunately, is quite limited in this regard.
So if this hypothesis about cytokinins and ABA, those are signals that roots are sending, they might still be different in those resistant ones, despite that obvious in terms of the strength of the root system is not quite obvious to you, but metabolically or something is definitely happening out there if you go by this kind of a hypothesis of cytokinins and ABA involved in this whole thing.
Yeah, basically, what's happening with resistance, I mean, the one, what's it called, 188, 788 resistance, am I getting that right? It's slowing down the cyst nematode infection. It's not stopping it completely. Mm-hmm. But the Peking resistance really shuts it down. So what you should see, right, the difference there would be, I would expect quite different root systems on those two types of resistances. I don't know if anybody's looked at that, but that would be a very logical thing to look at in terms of the fact that the Peking resistance just doesn't let the cyst get a foothold. It doesn't let it make the synthesia. Mm-hmm. So those roots should be quite healthy relative to the susceptible ones.
So Mark, I'd like to ask a question following up on that. So if we're gonna, evaluating roots is really changing apart from the chevelomics if they're in the field, but in general, people run away from that a lot because it's very time consuming and complex. Are there particular measures of root health that do you think are more better indicators of that health? And I'm thinking specifically of like when we use the rhizotron, you can measure the number of tips and branches and diameters and all this.
Yep.
Based on what you said, would it be a correct assumption that maybe looking at the measure of how many root tips would have a higher correlation with cytokinins and maybe a better indicator of potential differences in?
I like that basic concept. And people have done this. They've measured overall root growth. And it's number of apices by how big they are and how fast they're growing. That can be done in a rhizotron. I don't think it would be that difficult to do because you can do it on young plants. You don't have to grow really big plants to get that idea, get that concept down. In fact, I remember students working in agronomy looking at the very, very early stages of root proliferation and the branching off the primary nodes of soybean, and they could tell from those initial nodes how big that root system was going to be. So it's can be done in a very, I would say, controlled way. So you don't have to go crazy growing really big plants out in the field necessarily. So I would encourage that, absolutely, and include a little pathogen treatment on top of it would be very nice.
Are there any other questions for Mark? We're a little over 4:10, but if Mark's willing to stick around, we could probably take a few more questions.
Well, I absolutely encourage you to send me an email. I'm happy to discuss this other time, another way. Give me a phone call. That's my cell phone up there, by the way. And you're very welcome to call me. Happy to do it. Now, I will make one caveat. Hold on. This guy, oh dear. Oh God, my memory. Penny, yeah. This Penny character. He calls me every week. I love it. I absolutely love it that he would do that. So I encourage you, just call me. Happy to visit. You may be well beyond my my knowledge base, but please do.
Well, Thank you very much, Mark. With that very positive note, I think we can end our discussion. We really appreciate your time today and the insights you gave us. And
I appreciate that. Thank you very much.
Allison
That's really
clapping.
good.
See if I know how to do that. Let's see. Go reactions. Yeah, here we go.
Oh,
So much.
thanks so much.
Thanks,
Yep.
Mark.
Thank you, Mark.
Thank you.
Okay. How do you get out of here?
Bottom, the very bottom corner on the right should say end or end meeting.
Yeah, well, you may have to do it for me.
Okay, well, yep, well, mostly everybody has logged off. So Mark, thanks so much again. It was really interesting.
Thank you, Leonor.
You got my wheels turning now. Thinking of-
Good, let's do that. That's what you
Yes.
need.
Awesome. Well, thanks so much. Bye-bye. Thank you. And stop recording. Let's see.